Has anyone had an email about the stairs yet?
We haven't had an email, but the F4 doors are on today, so expect you to be late. They look shut. They look locked in any case, so.
So what does it say at the bottom of the stairs? Because I come from a different direction.
There's no entry, there's no way.
And you weren't told about this?
I changed it to no entry is for the lift or something like that. But,
Ah. So I did raise this yesterday, but it got as far as, no, we put up signs. So, oh, well, that's good. Did you actually tell anyone alternative routes? Did you give anyone any warning? Apparently it's going to be gone by Monday, allegedly. So by Monday, well, by Wednesday next week, you'll all be able to sprint up the stairs. Yeah. Unfettered, unhindered. I will give everyone a few minutes to make their way up, catch their breath, get used to the altitude. Whilst we wait, I'll give it a couple more minutes before I start today's lecture. But I could do a recap of yesterday, I can give some main points, but does anyone have any questions about yesterday's lecture before I start? Anything they would like clarifying? Not the whole lecture. No specific questions? Okay, so just to recap yesterday, we were looking at more of the traditional methodologies that are available for plant breeding. And realistically, we just have to appreciate this is a genetic exercise. You're attempting to breed two plants together, so obviously they have to be compatible. And realistically, you have to have a mechanism, a means of following traits through generations. And it's a genetic exercise, it's a mathematical exercise as well. So the more progeny you analyse, the more likely you will ultimately achieve at the product that you most desire. And the various means of doing this, so I talked about marker-assisted breeding, so that's very much dependent upon having good genetic maps for your particular crop of interest. But these have been developed over the last 50 years or so, and having a that map densely populated with molecular markers. So these are markers that are associated with particular varieties of the strain, so you will get different results depending upon the variety that you analyse. And this allows you to distinguish which part of the genome is coming from one parent versus the other. So it can allow you to delineate very finely, when you've done a cross, the insertion of the loci that you're interested in. And another point that I wanted to really hammer home is that from a plant breeding perspective, you're talking about traits. A trait is something that generates a phenotype that's desirable to you. You don't need to know anything about that from the mechanistic side. You don't need to know what the genes are. It might be multigenic, you don't need to know that. However, you do need to know where those traits map to. And that goes back to basic genetics. You can map things without knowing what the genes are, but normally in classical genetics, the purpose of mapping is to actually identify what the gene is. And then mutational breeding, we discussed about that, in that often what you're trying to do with mutational breeding is increase the number of alleles available to you, because traditional breeding is limited by the number of natural variants that exist. So mutational breeding increases that variability. And significantly, it's not classified as GM because there is no foreign DNA that is inserted into the genomes. Although, well, we'll see how this test case in France turns out, because that could have interesting implications. Sorry. And then I talked about tilling, which was utilising mutational breeding to then direct it towards specific genes. So in tilling, you have a specific gene in mind that you wish to mutate, and from a large population of mutated plants, you then work through those to identify plants that actually have a mutation in your gene of interest. And someone mentioned CRISPR at the end of the lecture. Why not bother just doing it with CRISPR? And we'll get to that in another lecture. But just to say, at this point in time, CRISPR is characterised in the European Union as GM. And we'll talk about that next week. And that brings with it its own limitations. And talking of GM, that's what we're going to talk about today. So much of the course involves the capabilities of transforming plants and analysing them. So today we're going to focus on how you actually generate transgenic plants. And I should say there are multiple ways to do this. We're only going to discuss a couple. But there are multiple ways to transform plants, but they all have their basic principles associated with them. So, just some of the learning outcomes for today. And I'm going to focus on really two examples today. So we're going to talk about Agrobacterium-mediated transformation, still one of the most popular ways of transforming plants, but particularly when plants were first being transformed in the late 80s, not all plants were amenable to this, and so other methods were developed as well. So we'll talk about direct transformation as well. And for some reason it's not showing on here. It should have been saying on this slide that this is extra reading, the pollen magnetoffection. So there's a possibility I've downloaded the wrong set of slides here. We will see. But there's some slides where we can look at this at the end. If I have downloaded the wrong one, don't worry. The right one is on mole. And in reality, all I've done is decide that there's a different endpoint to today's lecture, so I don't cover the magnetoffection. So, some of the points about making a genetically modified plant. So, three main steps, and I'm going to work mostly backwards through this actually. but you have to think about three main things, which is one, how are you going to introduce your construct? And actually there should be some stepping back at this point in time. But the next point is selection, and this is really critical. So how many of you in your course so far have transformed bacteria? How many of you know what the proportion of cells are transformed when you do a transformation? Low. We've had a low from the audience.
It's very low, like 0.1% or something like that.
We're going very low now, yes. So that is true of any transformation, and it applies to plants as well. And so what did you do in your bacterial practicals? When you're doing a transformation, what was the step that came afterwards?
Select.
Yeah, ooh.
Sorry, you go.
I guess just like selecting for them, so like
Yeah.
antibacterials.
Yeah. I mean, it is, but it's critical. The point I'm trying to hammer home is it is absolutely critical. Getting a construct into an organism has variable success rates, and so you do have to have some means of actually determining which of your population had taken up your construct and which of your population haven't. And yes, the vast majority will have not taken up your construct. So if you imagine that they have, then you're making an incorrect assumption, and you'll just be generating wild-type plants and wasting your time. So selection is absolutely critical. And the next point after that is actually regeneration of a whole plant. And I'm going to discuss this in a second, about the regeneration of a whole plant and what this means and the context of it. So let's start with going backwards through this. And the reason why I'm talking about regeneration. And this doesn't actually always necessarily apply in this context. And I'll mention what I mean about that in a moment. But most transformation procedures target the cell types that you see here. So there are various different cell types that can be transformed. But the point is often it is only a single cell that is being transformed. And a single cell is not a plant. So once you've transformed a single cell, you do have the issue of then having to reprogram its development and generate a whole plant from it. And there are various ways of doing this, but here we just show some pipelines for various different ones. So we have embryonic cultures. So here people have actually extracted embryos from maternal tissue. You can have organ culture. So in this instance, what they're trying to show here is they've extracted a part of the plant called the shoot apical meristem. Shoot apical meristems are regions of the plants that have meristematic tissue that can regenerate essentially all of the aerial parts of the plant. Now, callus culture, and we'll hear about this in a second, because this was one of the ways that plant transformation was done in the 80s, and it's still one of the predominant ways in which it's done. Callus culture, what you're attempting to do is take tissue and get it to de-differentiate into a mass of de-differentiated cells, fast-dividing cells that you can then culture and reprogram their development to regenerate a whole plant. And it's a little similar to cell culture, but in cell culture what you tend to do is take something like a leaf and use enzymes to digest away the cell wall. And what you end up with are things called protoplasts. So protoplasts are mostly what we call mesophyll cells. So for those of you who haven't really done any plant biology before, if you talk about the leaf, the mesophyll cells tend to be the photosynthetic cells within the leaf. But the point about all of these is that they then go through tissue culture procedures. And this is one of the bottlenecks, and has been the bottleneck for the regeneration of various different plants. So if you look back in the history of GM, you'll notice that certain plants were predominant in the literature. Things like tobacco, for example. And you might wonder why all of this literature was focused on tobacco. And the reason was, it was one of the first plants that people could transform easily and regenerate. It wasn't that anyone was particularly interested in generating transgenic tobacco as such, it's just that they could, and that was used as a model. And since then, people have applied these methodologies to numerous plants. But I should say that these procedures don't necessarily relate to genetic modification at all. Tissue culture of plant material is used worldwide to regenerate plants, particularly vulnerable plants. So it's a way of archiving tissue and ensuring that you can maintain that. Or there's certain aspects of the plant. So, for example, the shoot apical meristem is very rarely infected by viruses. And so if you want to regenerate plants that are virus-free, you often utilise the shoot apical meristem to regenerate plants via tissue culture, which is important in certain instances. So you might be having to regenerate from a particularly important plant species that has been affected by a virus, and you're attempting to regenerate that and ensure that the progeny aren't themselves just infected. But the end point of all of these tissue culture methods is hopefully a new adult plant. So what I'm going to do is take you through a basic pipeline for doing regeneration. So in this instance, we're going to start with some cells. This could be a leaf disc that you've punched out. They can do this with cotyledons. Cotyledons, if you see cotyledons mentioned in the literature, the cotyledons are the first embryonic leaves. So these are leaf-like materials that form within the embryo. And so they're embryonic leaves. So we take some tissue, and the first thing we're going to do is try and get it to de-differentiate, to start getting it to rapidly divide and producing a mass of de-differentiated cells. So this callus I was referring to. And the way you do this is you culture on media that is specific to your particular plant species. And you can see that there's an issue. You've got to identify what that media is, what the macronutrients, micronutrients balances are, whether they need to have certain sugars present, etc. And then the way you get them to de-differentiate is to introduce particular plant hormones into the media. And the two hormones that are commonly used are called auxin and cytokinin. So these, anyone come across auxin before? Yeah. So auxin was classically associated with Darwin's experiments with phototropism. So it's often involved, it's associated with expansion of cells, but along with cytokinin, it also has a role in regulating the cell cycle and also cell fate as well. And if you incubate cells on a mix of auxin and cytokinin, that often induces them to start to proliferate, and you form these de-differentiated calluses. Now obviously this isn't a whole plant, so how are you going to get to your whole plant? So the next stage after getting your de-differentiation step is to start to induce organogenesis. And this is normally achieved by altering this balance of auxin and cytokinin in the media. So you'll move your callus tissue onto media that now, if you're wanting to introduce shoots formation, will have an increased ratio of cytokinin to auxin. Sometimes you'll deplete the media of auxin completely, But generally, if you alter the balance in favour of cytokinin, that often will induce shoot regeneration from your callus tissue. And once you have that tissue, you excise it from any remaining callus, and to get it to root, you do the opposite. You change the auxin to cytokinin balance in favour of auxin, and this can then regenerate roots. So really that's a simple pipeline from going from a leaf punch through to regenerated plant material. But it misses out all of the steps that a tissue culture specialist have to perform in order to determine what are the correct conditions to do this for a particular plant species. And that is always the bottleneck, has always been the bottleneck for transformation. Getting DNA into cells, we can do that. Regenerating them is often the problem. And so you can think through a pipeline such as this, and some of the pipelines I showed in the previous slide, all of those points where you've got to make decisions and have possible variables. And of course the media is one of them. What I haven't said is that there are various different types of auxins and cytokinins. So you have to choose, you have to try out different auxins and cytokinins. You have to vary their concentration, determine what that is. And then you have environmental conditions as well. What lighting conditions are you going to use when you're doing this? What temperature are you going to do it at? So you can imagine this can become very much a box-ticking exercise of trying out numerous conditions, but it's very time-consuming. And it's, like I say, one of the reasons why certain plants were chosen as models for plant transformation at the beginning, because they were much easier to regenerate than others. So okay, that's regeneration. But now we need to think about selection. So as I said, when you're talking about selection, you're talking about a needle in a haystack. And just like with bacteria, bacterial transformation, yeast transformation, etc., the vast majority of selection procedures are involved with introducing a transgene, a selectable marker, into your construct alongside your gene of interest. And this primarily means then when you incubate your tissue on that particular agent, you kill off any untransformed cells. So here, the selectable markers that we're talking about often confer either antibiotic resistance. So these are going to be targeting either the eukaryotic or the prokaryotic ribosomes often, protein synthesis, or herbicides. And this is just showing you an example of a transformation where they've initiated organogenesis here. This is initiating callus proliferation here. And then once callus proliferation has happened on these leaf explants, they've then transformed them onto media that contains their selectable agent, probably an antibiotic in this case. And you can see that it's killed off most of the leaf discs apart from this one, which is actually starting to show some shoot material there. But just to go back to what sort of percentages might you be looking at, again, it does vary, but you're probably looking at around about 1% to 0.001% of cells being transformed. So that's what you're looking at in terms of transformation efficiencies. And I've just brought up a table here of potential selectable agents that you can use. And you can see you've probably come across things like kanamycin before. So you can use kanamycin for both bacteria and for plants. The gene that's involved is slightly different from the bacterial one. This is the MPT2 gene, which was derived from the TN5 transposon. But we have a range of potential selectable agents here. These are confer antibiotic resistance or herbicide resistance. There are some visible markers as well. So people have, for example, used GFP as a visible marker for selection. It only works really well in certain situations, particularly for something like Arabidopsis, where you can screen lots and lots of seeds because Arabidopsis has tiny seeds. It wouldn't be particularly good for example, using a visible marker for something like wheat, maize, etc. So we have a range of selectable markers, and I've just given you the genes involved, the enzymes that they encode, and where these have been sourced from. Now this is important. So you can see that often the sources are bacterial in origin. So is there anything you need to think about? So we've taken a gene from a bacterial origin and we're going to put it into plants. Is there anything anyone can think of that might need to be considered? Sorry, I'll start. Okay, so Ploid is one. Okay, sorry.
Infections, maybe?
Right, okay. So that's another.
Codon bias?
Codon bias, excellent. So there's all good answers, but the one that's primarily that you need to focus on is codon bias. Does anyone know what codon bias means? Has everyone come across codon bias? Well, if you haven't, if you analyse the sequence of various different organisms, determine what their codon usage is. So when they have a particular amino acid, and you have various codons that can be used for that amino acid, which codons do they prefer to use? And this relates to the tRNAs that they express. And you'll find that there is differences in the codon usage of bacterial genes versus plant genes. So if, for example, we took the MPT2 gene from the transposon and the tryptophan residues in that protein were all encoded by actually a tRNA that's very rare in plants, what you'll find is you'll never be able to get high expression of that in plants. So this will affect your selection. So for many of these things, once the sequence was known, they had to determine what the codon bias was and modify any amino acids and any codons in the sequence before they inserted into plants. And we'll hear an example of this in a later lecture, actually. But selection is absolutely crucial. You're going to come to do something later in this course where you have to think about constructs. And selection is one of the things that you really need to think about. So, that's going through two of the three main things. Now let's think about how we're going to get DNA into our plants. But before we do that, I just want to ask, so how we get DNA, this is all very practical at the moment, and we're missing out a sort of what I think is probably the most vital bit of it. If you're thinking about transforming a plant with a gene, I'll go back to my own research. Let's imagine I want to insert a copy of a regulator of stomatal development. What should I be thinking before I start any of this? What should I be thinking of before I start to make a GM plant? So that's a practical aspect. So it's part of the process. But before I even think about that,
What the result that I want to?
sorry?
What's the result that I want?
Absolutely, yeah. I should be thinking why I'm doing it. What am I expecting to get out of this? What's the purpose of doing it? So I'm going through the practicalities here, but ultimately there always has to be a why am I doing this in the first place? And that's what you should be thinking of in any of these examples. Why was this done in the first place? So let's move on to still probably the most popular mechanism of introducing DNA into plants was Agrobacterium-mediated transformation. So Agrobacterium It's a naturally occurring soil bacterium, and it causes something called crown gall disease. You may or may not have seen it. You may not have noticed what it was, but if you ever go buy some plants, often it's actually close to the ground. You see a big proliferation around the stem of the plant. So it just looks like it's got this mass at the base of the plant. And that's because Agrobacterium has infected the plant and introduced some of its own DNA. So this is an example of cross-kingdom transformation. Now they transfer a part of their DNA, apart from a plasmid, from something called the Ti plasmid. They transfer part of that Ti plasmid called the transfer DNA, the tDNA. And this contains various genes. And those genes induce the cells of the plant to divide rapidly, and also to start producing particular compounds called openes, which are amino acid derivatives, which are then used as a nutrient source for the bacteria. So the bacteria introducing this tDNA, it's making these galls, these tumours, and these tumours are transformed cells. Now, one of the problems for the plant biotechnologist is that, unfortunately, these tumours are really difficult to regenerate a plant from. And the reason is because of the genes that have been introduced, which we'll hear about in a second. But it's very difficult to work with this tissue, and often, if you can, the plants are infertile. So what's happened, and this was during the primarily the late 70s and 80s, was that bacterial geneticists, plant geneticists, disarmed these plasmids to make them useful for biotech applications. Now this is an overview of the process, and I want to make something very clear. If, for example, in an exam, you're talking about transformation, I want to make this super clear. I don't want to hear about this. It's not necessary to give all of this detail. I'm just illustrating the process here for a reason. This is what we call the Ti plasmid. So this is the bacterial plasmid found in Agrobacterium. And it has a region called the Vir region. This is the virulence region. It's the region that is required for infection, for transfer of the tDNA into plant cells. An important point to make about this is the Vir region is here. The DNA that's transferred into the plant cells, that integrates into the plant nucleus, the nuclear DNA, is here. This is the tDNA here. It's on the same plasmid, but it's not part of the Vir region. So it's acting in trans. They're not linked, except to be on the same plasmid. But that is something that can be easily resolved. So that's the key thing to take about it, is that these are not linked in any real sense. You don't have to have them on the same plasmid. So it's the tDNA that's transferred across into plant cells. And I should say that the tDNA can insert into the nuclear genome, and we don't really have any prediction about where it will insert. It's essentially random. There is an indication that it tends to go into more open chromatin, but that, of course, will be different depending upon which tissue that you transform. So the results are variable. You can't necessarily predict where it's going to insert. It's not like homologous recombination. So this is looking at the tDNA as was first found in one of these Thai plasmids. We can see that it encodes a couple of genes. And I talked about the fact that in the tumour, part of it is getting the cells to rapidly undergo cell division. And that's because the tDNA from Agrobacterium actually contains genes for auxin and cytokinin biosynthesis. So it's, in essence, doing something similar to when you attempt to produce callus from plants. Also has some genes that regulate the size of the tumour, and also ones that are required for producing these openes. What is required for transfer are these two components here, the left border and the right border. So these are near imperfect 24 base repeats. And they are needed by the genes in the virulence region to recognise that this is a tDNA and to mobilise it into plant material. Nothing in the middle here is required for that process. None of these genes are required. All you need is the left and right border. Now the Thai plasmid, the original Thai plasmids, are somewhere between 200 plus kilobases in size. These are huge plasmids. Bearing in mind a normal vector in E. coli is probably going to be something like 3 to 6 kb. So 200 kb, very unwieldy, very hard to work with, not easy to transform in, and certainly when you think about the tools that are available to people in the 1980s, very difficult to engineer. Because in 200 kb, it's very hard to find unique restriction enzyme sites. You know, I know a lot of things done today are done by things like Gibson cloning, etc. That didn't exist. This was all done by restriction endonucleases. So what they had to do was think about how they're going to disarm this. So, we've got to get rid of these tumour-producing genes. Okay? I talked about the importance of selection. So if you're going to introduce some DNA into plants, what you want to do is get rid of all this stuff that's not needed and replace it with a selectable marker gene. Now when I say a selectable marker gene, we're talking about something that's going to express. So we're talking about a cassette. It needs to have a promoter that's going to express in plant cells. It needs to have a transcriptional terminator as well. So think about your normal eukaryotic gene structure. Don't just say I'm going to put in a kanamycin gene. You need something that's going to drive the expression of it, and you're going to need something that terminates its expression. So the other side of it is the fact that the Vir genes are essentially operating in trans. So this plasmid is huge, it's very difficult to work with. You only need these parts, the left border and the right border, to start to engineer something. There's nothing to say that you need the Vir region and the tDNA on the same plasmid. So if you want to manipulate the tDNA region, the best option was to take that region and move it into a different plasmid that was much easier to manipulate. And in doing so, what you can then do is give it origins replication that will allow it to be propagated in both E. coli, so your standard E. coli, your standard bacterial strains for working in the lab, as well as Agrobacterium. So you're generating something that shuttles between E. coli and Agrobacterium. The Vir genes you leave on a plasmid in Agrobacterium. And what this generates is a binary system. So here we have a vector that contains the tDNA, and it's got a bacterial selectable marker because you're going to shuttle into E. coli. It'll have an E. coli origin, an agrobacterium origin. Your Vir plasmid with all your Vir genes stays in agrobacterium. So it'll only have an agrobacterium origin replication. Now this is sort of a first generation. It's still 20 kb in size. It was a large plasmid. Lots of work has been done since to make smaller, more manageable plasmids. And they've actually taken other components out of this plasmid and generated 3 plasmid systems. But the point is, you're generating a system that's much easier to work with in your standard practice, which is, working with E. coli. So I'm just going to do a typical plant construct. Promoters. Remember, this is going into a eukaryote, so you've got to have a promoter and you've got to have a transcriptional terminator for any part of the construct, any gene you're going to express, whether it's the selectable marker gene or your gene of interest. So you need a promoter. I'll put here the 35S promoter. This comes from the cauliflower mosaic virus. It's often used to generate high-level expression, but you've got to think, is that what you always want? You might want to use a tissue-specific promoter so that it's only expressed in particular tissues or at particular times. You could use an inducible promoter. So you've got lots of variability to think about there. You've got your DNA that you can express. So again, it comes back to what are you trying to do here? What are you achieving? So you've got to think about what the gene is and what it's going to do, and importantly, how are you going to assay for it? Think about that. How are you going to actually tell if your construct has given the trait to your plant that you actually want? You've got your terminators, so these are things that ensure that you don't get transcriptional read-through and that you get then poly-A tails added to your proteins. If you don't have that, your protein will just won't be actually translated, it'll just be turned over, so your gene won't get any expression. And of course the selectable marker, and the same rules apply to this. And finally your left and right borders. They're absolutely required for the transfer process. Once you've actually generated your plasmid, done all your cloning in E. coli, you shuttle it into agrobacterium with your Vir plasmid, and then you infect your plants. And this is very much dependent upon the plant species. Often it's leaf-X plants, so where you've just punched a piece of leaf material out of a leaf. You can also do this with roots as well. There always has to be a wound though. You always have to generate a wound that the agrobacterium can then access plant cells via. And this stimulates expression of the Vir genes and transfer of the DNA. And then we go through the processes that we've just seen to transform and to select for those transformations and then regenerate. Of course, then everything happens by standard genetics from that point on. As long as they are nuclear encoded, you can just rely on your standard genetics to ensure that they're going to get transformed to the next generation. Now, this isn't in the new one, so I'm just going to go past that. The last 5-10 minutes, and if I don't cover it, I'll cover it in the next lecture. I'm going to talk about direct gene transfer. So, Agrobacterium is great, but some plant species are recalcitrant to Agrobacterium infection, particularly actually the major crops, things like wheat. So, other systems have to be developed. And if you think about transformation in E. coli and things like that, how do you do it? You can use electroporation, you can use chemical methods to permeabilize the cell. In plants, they've got a hard cell wall on the outside, so those methods don't work particularly well unless you're working with protoplasts. So the other way of getting DNA into cells is to essentially coat a cannonball and fire that cannonball into your plant cell. But just imagine making this all a lot smaller. So that's where we talk about direct gene transfer and gene guns. So the principle here is relatively simple. You take metal nanoparticles, often it's gold. The best results often do come with gold because gold actually forms nice spheres, whereas something like tungsten, which is also often used, the particles are a lot more irregular, a lot more spiky. They cause more damage when they go into the cell, which means that actually you kill more cells than you transform. But what you essentially do is you take nanoparticles that are, 1 micron, 5 micron, 10 microns in size, and you put your DNA onto them, the DNA that you want to introduce into your plants. Now, I want to make something clear here. These constructs, the same rules of what I've just mentioned about a construct previously apply, in the sense that you are introducing DNA into a plant cell. So they've got to have a promoter, you've got to have your gene of interest, terminator, you have to have a selectable marker gene. But you're not using agrobacterium here, so you don't need the tDNA repeats, you don't need any of that. You don't need a plasmid, you can do this with linear DNA, but the same principles apply. Your gene with its regulatory sequences and your selectable marker. And then what a gene gun does is essentially it uses gas, high pressure gas, to accelerate these particles and fires them into your plant material. And so they basically break through the plant cell wall, and some of them will go into the nucleus, and the DNA will then integrate into the genome. And the fact is, we don't know too much about this process. We just know that it happens. Plants, the nuclear genome at least, does not do homologous recombination. So, the DNA can go in, and actually, the problem with direct transformation like this is often you can get multiple copies going into the genome. You can get all sorts of rearrangements as well, so it's not actually as directed in terms of the structure of your construct at the end of the day in the genome is not as predictable as what it would be with agrobacterium. So that's one of the issues. But it is a way of getting DNA into plant cells. And you can target various plant cells, but you still have to retransform afterwards. So you still have the same problems with regeneration. So last couple of minutes, and I'll go over this again at the beginning of the next lecture, is the other outcome that's really useful for plant biotechnologists. Using this mechanism, you don't just have to target the nuclear genome. Plant cells generally have another genome, more than one genome in fact. They have mitochondrial genome, but they also have the chloroplast genome. And the chloroplast is a useful little target. The reason: the chloroplast is not inherited via pollen, it's maternally inherited. And this is really important if you're thinking about GM plants and controlling the spread of GM plants. One of the issues that trials always have is how do you contain pollen, GM pollen. And one of the ways you can do it is ensure that no GM material ever makes it into the pollen. And if you transform the chloroplast, that's what you do. You ensure that you're never going to get GM material in pollen, because the chloroplast is maternally inherited. The other advantage that you have is that, unlike the nuclear genome, for the chloroplast, you can use homologous recombination. And the reason is the chloroplast is derived from cyanobacteria. It's derived from a prokaryote. So homologous recombination means that you would modify your construct. You'd have the ends of your construct designed so that they would homologously combine with a section of the bacterial, the chloroplast genome. So you're going to know where your construct should be inserting. And the other thing is there are multiple chloroplasts per cell and multiple chloroplast genomes per chloroplast. That means you are getting lots and lots and lots and lots and lots and lots of copies, which means you can generate really high expression, much higher expression than you would do if you focused on the nuclear genome. So if you want high expression, then it's often a good idea to target the chloroplast. The only caveats that you have to bear in mind is that this is a prokaryotic system. So your promoters have to be prokaryotic. They have to be recognized by a prokaryotic RNA polymerase. If you want to translate it, you have to have a Shane Diagano sequence in your 5' UTR. And your selectable marker gene has to be one that is functional in the chloroplast. So the one I mentioned here is AADA, which gives you resistance to spectramycin, which targets the chloroplast 70S ribosome. And finally, the final thing I'll say today is that multiple genomes does create its problems because actually the same rules apply with this as to any transformation. You actually only transform a small number of these genomes. So even within a chloroplast, you might actually only have 1 out of 50 genomes transformed within a single chloroplast. You want to generate a plant that is what we call homoplasmic. All of the genomes are transformed in every chloroplast in that plant. You start off with a plant where actually very few are. So what you have to do is you have to maintain selection over several generations to ensure that eventually all of your chloroplast genomes are transgenic. Okay, I will go back over this part of the lecture in our next lecture. But for then, if you have any questions, drop me a line. Happy to answer them. If you do have questions, what I tend to do is I'll put them up so that everyone can see. But I'll obviously anonymise so that nobody knows who's asked the question in the first place. But I will be here next week, just to be clear. So I'm not striking, so I will be here.